Now we are going to take into account endocytosis and exocytosis. Break the terms. Cyto, that means it has something to do with cell. Okay. Endo means inside. So something inside the cell, some mechanism or some process which has something to do with inside the cell. That means cytoplasm we are talking about. Exocytosis means outside the cell, and because we are discussing the membrane transport over here, so we are dealing with the movement of the particles across the cell. Alright. Now first thing you need to get aware about is a term known as vesicle. What is a vesicle? Vesicles are very important for the transport purpose, not just across the cell cytoplasm and outer atmosphere of the cell. They're very important for organelle transport as well. All right, the transport of the material inside the cell or outside the cell is being conducted by the vesicles which are formed inside the cell. And as you can see over here, how a vesicle is being formed. Let's take into account that something has to come inside the cell. All right, some particles have to come inside the cell. Now over here you might think one thing, that where are the carrier proteins now? Now we are dealing with formation of vesicles. Certain things, as you understand what lysosomes are, there are certain things which are responsible for bulk movement and they would be carried out with the help of vesicles. This is a vesicle basically. As you can see, this vesicle is not present anywhere in the beginning of endocytosis. We are taking into account endocytosis over here. That means few particles would come inside the cell from outside the cell. There are many particles. They would be engulfed by the plasma membrane. This is nothing but plasma membrane, the lipid bilayer. These are the lipid heads. These are the tails, both sides. This membrane would encircle, or it would form a protrusion inside the cytosol. As you can see, this protrusion is formed inside the cytosol. It would go further and further, it would deepen and it would separate. That means it would nip off, leading. That's why we say that the fluid mosaic model has to be dynamic, because all these things, if it is not dynamic, they cannot be conducted. So this vesicle gets separated. The lipid membrane, it forms the vesicle. Alright, the vesicle is also having lipid bilayer. It forms the vesicle around the substances, and later on these substances, wherever they are required inside the cell, they would move over to there. This lipid membrane would be digested by the cell machinery of the enzymes, and the particles which had to enter inside the cell, they would be used up by the cell. Alright, this is what is known as endocytosis. Consider that you require few ions. Ions would be transported across the membrane. We do talk about ion transport, that is they easily go through the carrier proteins, or they go across the membrane with the help of ATP, or they at times do simple passive diffusion. But if you're talking about large biomolecules that have to pass across, and other biomolecules which are required in great amount by the cell, let's talk about amino acids have to enter the cell, they would be entering by the process of endocytosis. Alright, now something inside the cell is toxic, or it is not required inside the cell, that has to be thrown out of the cell. Alright, that is to be thrown out by the process of exocytosis. Something inside the cell which is not required by the cell would be engulfed by the lysosome, or the vesicle which would be formed by that lysosome. That vesicle would come and try to merge with the plasma membrane. The plasma membrane, supposedly, try and understand it this way, that supposedly this is the plasma membrane, see over here. Now this vesicle would come and try to, this is the vesicle, it comes over here, it comes and merges with this. So according to the fluid nature, this membrane would become like this for some time, and whatever particles are present inside it, this membrane is fluid. So it elongates, or it folds itself, or it embeds all the lipids which were present in the vesicle, and the particles which are not required, they would be thrown out by the process of exocytosis. Okay. There are other terms like phagocytosis and pinocytosis. You might have heard about them. Phagocytosis is a very common term that we hear about in immune system of our body. When we say that the immune cells of our body, they do phagocytosis of particular foreign bodies and help us in protecting from some sort of disease. That process of phagocytosis, supposedly this is the particle that is outside of the foreign body. The membrane would come and form like this, and then there would be many enzymes which would digest it. Okay, that is engulfing of the foreign material, that is known as phagocytosis. Same way, it would be thrown out of the cell in similar mechanism that we have understood over here. And this is, phago is eating and pino is for fluid substances. Supposedly it is water, supposedly it is any other constituent which is present in fluid form. The taking up by the cell would be known as pinocytosis. Taking up by the cell, that is the food particle, that is a particle being eaten up or engulfed, that would be phagocytosis, and that in the case of water would be known as pinocytosis. So this is all we have to understand about endocytosis and exocytosis. Of course, there is membrane transport, that is diffusion transport taking place, but at the same time there are certain particles which have to be thrown out in these ways. So for that we understand endocytosis and exocytosis, and this is all we need to understand about the membrane transport across the cell. Okay, thank you.